Hi, my name is Fang, and I'm a PhD student at Chongqing Medical University in China and a visiting student at the University of North Dakota. And today I will talk about scGEN, leveraging adaptive waiting strategy for changing cell clustering in single-cell RNA-seq. First, I want to give a short background. Unlike traditional bulk RNA-seq, not only provide an aberrant gene expression profile from a mixed cell population, single-cell RNA-seq captures information from individual cells and revealing the heterogeneity. The key features of single-cell data are high sparsity and complex nonlinear gene interactions that need specialized method to reveal the true biologic heterogeneity. We all know single-cell clustering is crucial for downstream analysis. The main challenge in single-cell clustering is accurately classifying cells into distinct cell type or state, especially for cell lines in the boundary regions. Traditional methods such as K-means cannot handle this complex problem, and while deep learning methods have shown development, they still struggle with this difficult to classify cells. So to address this challenge, we have developed scGEN, an autoencoder-based algorithm that's specifically designed to improve clustering accuracy for these difficult to classify cells. Our approach consists of three components. First, we extract features from both attribute and structure metrics using dual neural networks. Second, by calculating attribute similarity and structure similarity, we create a comprehensive representation of cell relationships across multiple dimensions. Finally, we introduce an adaptive weighting mechanism using improved ZNB loss combined with hard sample loss. In summary, this novel approach guides the network to allocate additional attentions to cell in boundary regions. Next, I will introduce our scGEN step by step. We begin with the gene expression profile as our input data. First, we calculate the Pearson correlation between each cell pair. Sorry. Sorry, can you see my screen now? So we begin with gene expression profile as our input data. First, we calculate Pearson correlation distance between each cell pair. And for each cell, we rank the correlation in each row to identify the case largest value as a cutoff. So here we set the k equals 10 as a common baseline in single-cell analysis. And we will plan to test the different k values in the future version. So the next step is to calculate the adjacency matrix. For the adjacency matrix, we set all Pearson correlation greater or equal to this cutoff as 1 and others as 0. As a special note, the diagonal values representing the correlation between a cell and itself uniformly set to 0. So based on this adjacency matrix, we generate a graph representing cell relationships. And we propose that if cell x1 is adjacency to x2 and x2 is adjacency to x3, this means that x1 and x3 may share a certain degree of similarity. We then use two parallel neural network to independently extract features from the attribute and the structure matrix. Although these networks share identical architectures, they use separate weight, allowing the network to improve their ability to capture distinct information dimensions. The next step is to integrate these two types of features. So we calculate the joint similarity that combines both attribute similarity and the structure similarity. And we use the hyperparameter beta to balance these two types of information. And we also generate a joint representation that integrates the attribute and the structure features. So strategies ensure scGEN effectively capture both attribute and the structure information. The next step is guide the network to focus on hard samples. First, k-means clustering is performed on the joint representation matrix to generate the initial clustering result, which generates pseudo-label for each cell. And then we calculate the distance of each cell to the center, to a cluster center, and the top n sample close to the center are selected as a reliable set because their classification is more certain. So for each cell pair, if they have the same pseudo-label, for example, both belongs to cluster 1, this indicates a positive pair, and otherwise we denote them as a negative pair. And the next step is to assign weight to each sample pair. In scGEN, we have two loss functions. The hard sample loss function effectively leverage attribute and structure information from the cellular matrix, and improve their NB loss is used to prevent overfitting. And these two loss functions are combined to improve clustering result. And the hyperparameter alpha is used to balance the contribution of these two loss functions. Here are the data sets used in our study, including both real data and simulated data. So real data includes both human and mouse, and the sample size ranged from 200 to about 6,000 cells. The simulated data includes up to 16 populations. We first compare our scGEN with 7 published methods across 8 datasets using 3 metrics. It's very clear that scGEN outperforms all the 7 published methods across all 3 metrics. And our scGEN has 2 parameters. Alpha balances the contribution of the two loss function, and gamma adjusts the attention weight assigned to hard samples in the learning process. So after exploring a comprehensive parameter space, we discovered that the performance remains stable across most datasets. So this demonstrating that scGEN's inherent robustness without require extensive parameter tuning. To assess the contribution of each loss function to overall clustering performance, we carried out 3 variants: the completed scGEN and scGEN without the NB loss, and scGEN with hard sample loss. And the results show that the integration of these two loss functions significantly boosts the scGEN's clustering performance. And next, we generate synthetic data to check how well scGEN handle complex differentiation trajectories. And where all methods show reduce with the trajectory increase, the scGEN keep the high accuracy, and scGEN decrease is far more gradual. So this demonstrating scGEN's superior ability to classify these difficult to classify cells. So both real and synthetic data show scGEN gives more accurate clustering results, which will help later biological analysis. So to check this, we reanalyze the published dataset. This is a human fetal pituitary spanning 7 to 25 weeks post-fertilization. So we identified 40 cell types, including stem cell and other differentiated cell types. We think if the cell type obtained by the algorithm are consistent with the original paper, we mark them as consistent cells. Otherwise, we think they are inconsistent. So this plot shows that scGEN achieved the highest consistency with the original paper. However, among these In inconsistent cells, the most notable differences is the stem cell. Stem cell classified as in the original paper were classified as polygonal cells in scGEN. The hypothesis is that if two cell populations have fewer differentially expressed genes, they should be more similar. So we compare the consistent polygonal cell versus consistent stem cell, inconsistent stem cell versus consistent stem cell, inconsistent stem cell versus consistent polygonal cell. We can see that the first group has the most differentially expressed genes because they are totally different cell types. But the third group has the fewest different expression genes, so they suggest these inconsistent stem cells are more likely to be polygonal cells rather than the stem cells. So to further check this result, we check the direction of these shared DEGs. As expected, all showed consistent directional changes. So this further supports that our scGEN's classification and that is this inconsistent stem cells should be the polygonal cells. And the geo-enrichment analysis also support our result. The stem cell development was downregulated and the positive regulation of separation was upregulated. So to give you a summary, that scGEN improved the clustering result, and scGEN enhances cell type identification, and scGEN maybe enables integration of spatial and temporal insight. Thanks to my mentor and all members in my laboratory. Thank you. Any questions? Sorry, I cannot hear you now.
Can you hear us now?
Yes.
Hi. Yeah, thank you for your talk, and just want to let you know you get a big round of applause if you didn't hear it. And we do have a Q&A. Can you see the Q&A tab in your browser? Yeah, next to the chat box. If you can see the Q&A, yes. So that if you scroll up or down, yeah, this is the question. Oh, this. Yep. Yes. I think you're seeing this question from about this. Are you seeing the question, Fang? How are the testing set is obtained?
This question?
Yes, if you can answer that one.
Testing.
How are the testing set is obtained using which method, and how is the clustering done to attain the ground truth labels that are in turn used, I assume, to assess the more accurate clustering methods, of which scGEN achieves the highest accuracy, etc.
Oh, the testing data is obtained just from the published paper with the true labels.